Before we get started on our lesson on the plasma membrane, I just really like this cartoon. I find it hilarious because notice you got the hydrophobic areas of the cell membrane shying away from that water gun because remember, they're phobic of water, they're scared of it. So let's talk about what makes up the cell membrane. The biggest area by far, the most important molecule in the cell membrane is a phospholipid. A phospholipid has two fatty acid chains and then a phosphate group attached to a glycerol. So it's got some similarities to the structure of triglycerides, which we've talked about in our lesson on lipids. Phospholipids are very special because they have both hydrophilic and hydrophobic regions. They have a polar phosphate head, which is hydrophilic. Hydrophilic means water loving. So those are the areas I've kind of highlighted in yellow. And then they have a set of non-polar fatty acid tails, which are hydrophobic or water hating. And I've kind of highlighted those in purple here. And keep note of those colors because we're going to keep labeling things that are hydrophilic yellow and things that are hydrophobic purple. So the cool thing about phospholipids is that when you stick them into water, they can spontaneously form structures like what you see here. It's not important to know what they're called because you honestly don't see structures like this in cells, but they form the kind of basic structure on which cell membranes, plasma membranes, all membranes in the cell are made. And what happens when you put a bunch of phospholipids in water is they form a phospholipid bilayer. So bi means two. So there are two layers of phospholipids facing each other. Some people also call this a phospholipid sandwich. I want to point out that this phospholipid bilayer, this kind of sphere of phospholipids, is found in surrounded by an aqueous solution. And remember, aqueous means it's a water-based solution. And the cool thing about this is you can also have an aqueous solution inside of this phospholipid bilayer sphere as well, which means you can have two aqueous solutions separated by a barrier. And the properties of this barrier are going to be important. So let's look at what's hydrophobic and what's hydrophilic here. So you have polar hydrophilic heads that are facing either towards an external aqueous solution, like what you see here, or towards an internal aqueous solution. So notice that the polar heads are always facing a watery solution because they're polar, they're attracted to the polar parts of water molecules. Then you have a non-polar kind of hydrophobic region in the interior, in the center of that sandwich. And that's going to be really important to the functioning of these phospholipid bilayers. They form a kind of barrier that is very particular about what types of molecules can cross. So if you have something that's polar or charged, so let's say we have an ion, and it can interact with the polar heads of these phospholipids. It cannot, however, go any further. So it is blocked at this point because it will not be interacting with the non-polar tails. So for example, something very charged is going to find it hard to make it through this barrier. So this allows you to separate and then change the solution that's inside of this bubble compared to what's outside. And that allows you to have a different internal environment inside your cells from the environment outside of your cells. Allows you to concentrate solutions, control what enters and leaves, and that's what makes cells and therefore life as we know it possible. So there is more than just phospholipids in a cell membrane. Some important molecules that we're going to talk about are the embedded proteins. So the embedded proteins are stuck or embedded within the cell membrane. They're kind of stuck in it. I don't know what other way to say it. They have hydrophobic and hydrophilic parts as well. And what happens is the hydrophilic regions, and keep in mind, same highlighting here. If it's yellow, it's hydrophilic. Notice that you have aqueous solution out here. You have an aqueous solution in here, and the parts that are facing the aqueous solution are hydrophilic because they need to be able to interact with water. You could also, if you really wanted, you could put a little hydrophilic area in the center of this protein. However, the part that's exposed to these fatty acid tails has to be non-polar because it would be excluded by water. If it was polar, it would exclude itself from the inside of that membrane, and you would end up having that protein like pop out. So in the any proteins that are in the cell membrane or are embedded in the cell membrane, you'll usually see a hydrophobic section that's going to be stuck in that region with the hydrophobic tails. Then you do also have cholesterol molecules that are embedded. They have an important role. They do have a hydrophilic and a hydrophobic section that allows them to nestle in with the phospholipids. So you see that on the right as they are arranged in a cell membrane. Notice that you've got some blue over here. That's representing the water or the aqueous solution. You got the polar part facing it, the non-polar part facing inside the cell. And the most important thing about cholesterols or other steroids are that they add membrane fluidity and they help stabilize it. So they keep these phospholipids. Notice how these phospholipids are really tightly packed together. They keep them separate from each other so that way there's some movement allowed. And then also they help when it's warmer in temperature, like if you're getting a fever and your body temperature is 102 or something, they'll help make sure that your cell membranes don't start to come apart because they get too fluid. So that's what's happening there. And then in cold temperatures, they're going to keep your cell membranes able to move and be fluid, even though it's getting colder, because they're going to prevent these phospholipids from tightly packing together. Oops. Come on. So let's talk about what happens when you put all these things together. So there are a number of molecules, like I said, that are embedded in the phospholipid bilayer or make up a part of it. So there are proteins. There are two major categories. Integral proteins are actually stuck into the membrane. They can either cross the entire membrane or they can just be partially embedded, and they can have various functions. So some of them serve as channels, some of them serve as receptors, some of them are enzymes, some of them are transporters, and don't worry, we will talk about each of these types of molecules by the time we get done with the year. Then there are peripheral proteins that are attached just to one surface or the other of the membrane. They can be attached to the cytoplasmic side, or they can be attached on the extracellular side, which is outside of the cell. Because remember, there are two surfaces of a cell membrane. These guys can serve as anchors for parts of the cytoskeleton. They can serve as cell adhesion molecules, which help cells stick together. They could also just be enzymes. We'll talk about some of those later. Then you have those steroids, which in vertebrate animals like ourselves are cholesterol, which we talked about on a previous slide. You have glycoproteins. A glycoprotein is a protein with glyco, meaning sugar, attached to it. So basically it's a hybrid molecule that's part protein, part carbohydrate. And these guys can have various functions as well. Some of them are for cell signaling or cell recognition. So that way your body knows, hey, this cell belongs in our body. Some of them are immune factors. Some of them serve as structural anchors to help keep cells together. You can also have glycolipids. So that prefix glyco means sugar. Almost means glucose, but not necessarily. And glycolipids are lipids, kind of like the phospholipids, but instead of having a phosphate group on them, they have a carbohydrate chain. These guys help to stabilize membranes. They can also serve as some receptors. And just to show you what these guys look like. So here we have a glycoprotein. Notice it's got this chain of they look kind of like beads here, but honestly, they should have made them like hexagons or pentagons. Here you have a glycolipid. Notice it doesn't look like it's attached to anything like one of these blue proteins. That's because it's going to be attached to the heads of one of these phospholipids here. Then you have the phospholipids. You also have membrane proteins. So this is an integral protein. Notice it's stuck through the membrane. This is a really good example of a peripheral protein. It's just kind of floating on the top. It doesn't necessarily go into the membrane. And then here we see some cholesterol. So when we put all this together, scientists have come up with a model for how the cell membranes work, and it's called the fluid mosaic model. The mosaic is the fact that there are a bunch of different molecules making this up: proteins, cholesterols, phospholipids, etc. And the fluid part is that molecules can move around along the surface of the membrane. So I think of it as they move in 2D. So molecules are not going to move up out of the cell or down into the cell. Not if they're stuck in the plasma membrane. They're going to be staying there. But they can move side to side. They can move back and forth. So anywhere within the two-dimensional plane of the membrane, they can kind of float around. That's why you see like this kind of peach-colored protein that's sitting here. This peach-colored protein is right there. It is just kind of floating along. And that membrane fluidity can be important for various things. One thing we're going to talk about is that the membrane doesn't necessarily stay in place. It can pinch off and form little vesicles. And that membrane fluidity is important for that. Okay, if I can get this to move on to the next slide. Let's see. Okay, I do not know what this is doing. Let's see. Can I get this to move on? I don't think I can. Luckily for us, this was our last slide. So we're going to go ahead and end here.